Okay, reaction rate lab. So I handed out a reaction rate lab, and you're going to be performing your own experiment with this. And I just thought I'd go through maybe my thought process and how to set up this lab and work through it, and maybe just give you some ideas about things you can be doing. One thing when we're doing a lab in our investigation, we have an independent variable, and in this case, it's really what we're trying to test. So we're looking at some factors that affect reaction rate, like temperature or concentration or surface area or a catalyst. And in order to see how fast those things affect reaction rate is really measured by looking at time. The longer the time, the slower the rate. The less the time, the faster the rate of the reaction. So for everybody, the dependent variable is going to be the time. It depends on the temperature, or it depends on the concentration, or the surface area, or whether you added a catalyst in doing that. So what I did was I chose a reaction that I'm going to do, and I'm going to first try to mess with temperature on this. And this reaction is a little bit of a complex reaction, but I take a clear solution of I minus. It's actually potassium iodide, but the potassium is a spectator ion. And this is ammonium, which is a spectator, and then this persulfate ion right here, kind of a complex ion mix that we have. And this is also a clear solution. I did lace this with a little bit of starch, and what happens is starch turns a dark bluish black in the if it's in the I2 form. Not in this form, but in this form right here. So I mixed in a little bit of starch with this right here. It's still clear. And then this will be another byproduct. So in this reaction, I'm going to actually be able to not gradually see a reaction, but it's going to be nothing, and all of a sudden, bam, it'll turn dark blue, and then I can look at these variables and measure it that way. Some other reactions that you might do might be a more gradual thing, but there are still ways you can keep track of that and see how factors affect reaction rate. So I'm looking at this, I got a thermometer here, and it looks like it's about 19 degrees Celsius. I'll assume these both are the same, and what I'm going to do is take these two things, I'm going to combine them in here, I'm going to start recording the time on this, and let's see, 1, 2, 3, go. I'm going to add those in there. It looks like it kind of turned a color, and okay, we're at about 5 seconds, 10 seconds, 15 seconds, 20 seconds, 25 seconds, 30 seconds. Oh, it looks like it's about 37 seconds it turned blue. So 37 seconds it turned this dark black color, and you can definitely see what happened there. Now I have some things that I just heated up, okay? And this has been a little bit hotter, but it's about 39 degrees Celsius right now, okay? And what I'm going to do is dump these two things together right here. So it looks like I have the same amounts of each reactant. Everything's the same, except I'm changing one thing, and that's the temperature. I'm going to dump it in right now. Okay, I'm going to watch what's happening to it. And let's see, 5 seconds. 10 seconds. 15 seconds. 20 seconds. 25 seconds. 30 seconds. Okay, 31 seconds for this one. So this one went in 31 seconds. It was a little bit faster. I think the first one was 37. The next one was 31 seconds. Okay. Now, if I were doing this, I would try to get at least 4 data points. So I might do one at 20 degrees and 30 degrees and 40 degrees and 50 degrees, maybe 10 and 0 degrees. try to get a nice spread of data points and see if I can see a trend or pattern. Now let's say you don't have a thermometer at home. Could you still do a reaction like this? Sure, you could do ice water, room temperature, lukewarm, and then hot, and that would work as well. You'd have more observational data for that, and you can always get your time data for that right there. But that would be an example of comparing temperature and time. Now I'm going to go to another experiment, and this one I'm going to look at the concentration differences. So I'm going to start with these two amounts and put them in here, and let's see, 3, 2, 1, go. Okay, notice that color change before they got mixed in. It dropped back out. Okay, and now we're about 10 seconds, 15 seconds, 20 seconds, 25 seconds. Okay, that one's about 30 seconds, okay? So that one was right there at 30 seconds. A little bit different amount than in these two, so I'm kind of doing a whole new comparison set with what's going on. And I think we said that was about 30 seconds. Now I'm going to do the same thing, except what happens is this solution right here is going to be the same, but this solution, I'm going to cut the concentrations in half. So instead of putting 20 milliliters in there like each one, I have 10 milliliters of this substance and 10 milliliters of water. So the total volume is going to be the same, but there's less active ingredient. This one's concentration got cut in half. I didn't mess with this one. I kind of want to only mess at one thing at a time when I'm doing an experiment, okay? But I could have just as easily messed with that as well. Okay, so I got my two things. I'm going to dump them in here, and 3, 2, 1, go. And I'm going to swirl that around. Okay, 5, 10, 15, 20, 25, 30, 35, 40, 45, 50, 55. Oh, it's not working. 60 seconds. 65, 70, 75, 80, we're getting close to 90. I'm wondering if I put the right things in. I might have to do a quick double check. Here we go. We got it. What, 95 seconds. Notice it's not quite as intense because it's diluted down. So that one took quite a bit longer by diluting it, and it seemed to have a pretty large effect. So again, concentration is one of those things. You might not have milliliter containers or different types of things. I could do it in moles per liter, but I could simplify it, keep everything the same. And in one I had 10 milliliters of substance, this substance here, and 10 milliliters of water. One I had 20 milliliters of this and 0 milliliters of water. So you could keep proportions and ratios and keep track of that and get data points to do measurable data with that, whether you did concentration or just held everything the same and did volume, or everything the same and did mass, or something like that. So I just showed, again, a couple of different experiments, one showing temperature versus time. Time is a dependent variable, temperature is independent variable, and again you get 4, 5, 6 data points for this, and then concentration, you get 4, 5, 6 data points for doing that. You You could also use a surface area lab or a catalyst lab, or even a nature of reactants lab if you chose to do that. Any questions as you're doing the lab or about your approach, or if it seems like a good idea to do, I'd certainly be glad to talk with you about that. And good luck. Enjoy your lab project. Thank you.